Testing one, two, three. Hey, good evening. I think we're live again. Friday, March 12th, 2021. Oh, a year into the COVID. Yeah, well, guess what? Good. I see you down there, Luna. Hang, hang, hang on a minute here. Um, no, tonight I'm excited about a guest. We're having a little bit of technical difficulty, and I wasn't quite sure she'd pop on, but I see Luna Whitcomb. She's a cannabis advocate, and I'm looking forward to hearing her story. And without much further ado, I don't have a whole lot to say this evening, but I'm excited. I'm excited. This is someone I don't think I've paths have come across, at least not knowingly. But anyways, Luna, are you ready to come on up? Give me your. Well, we'll be testing on air live. Can you hear me? And this might not work too. I don't know. I think I'm going to try to remove you. I'm not sure if you can hear me. These things happen. These things happen. We're getting some buffering if you can hear me, because your thing kind of locked up. I'm going to remove you and try to come in again, please. We'll give that a try. Sometimes that works. Well, technology, what you get, what you pay for, right? And stuff is pretty much free, so I guess that we get. But maybe we'll have a guest. I mean, usually there's always technical difficulties. That seems to be par for the course. And anyways, you gotta learn to go with the flow, right? So what's going on, guys? Got any topics? We can do open panel for a minute until we get this thing straightened out. But about time for planning. Actually, I wanted to get some opinion on when's a good time to start your seeds, and I guess the time would probably be already. They should be started. But would like to hear some of your thoughts. I actually haven't started yet. I was thinking a lot about it this week. Just at least starting them indoors. I read somewhere where you want to wait till at least the equinox, which is going to be in about 8 days. Welcome, Shepherd's Homestead and John. Good to see you. Wildman, I thought you were going to get a hold of me a week or so ago with a text. I never heard back from you. Top Floor Genetics, welcome to the show. Cloud 99. I saw your thing about Oklahoma City. May's kind of a tough. May's the peak month usually, so it's real hard for me to get away. Good to see you, Cass. Oh, we might have another opportunity here. Let's give it another try. Yeah, I think that's working better.
Can you hear me? I can hear you.
Perfect. Oh, excellent. How are you doing?
I'm good. How are you?
Great, and thank you so much for your time, by the way. I know time is what we have, and you're sharing it with us, so I appreciate it.
Yeah, absolutely. Thank you for having me on your show.
You are welcome.
Yeah.
So I didn't get a chance to go over. Usually I'll go over things like, is there anything you can't talk about or don't want to talk about? So I'm just going to let you, your biography is pretty good. Skunk Magazine, and I know you're in some forums. You're a moderator. Tell us a little bit about. Would you mind telling us a little bit about? I don't know you that well, and I'm looking forward to.
Yeah, sure. So jumped in the community, about 12 years ago. Just kind of made myself really active in like the forum scene and online on Facebook and stuff like that, trying to spread organic and probiotic and natural farming methods to people. Yeah, just really big into different, soil food web. I like to experiment a lot with microscopy, playing with different food sources, different approaches, trying to maximize nutrient uptake, things like that. So I have some pretty cool things I'd like to talk about too.
Oh, that's okay. Can I go off on some tangents once in a while?
Yeah, let's discuss some cool stuff.
You know what got me kind of started, I have a tea mix that I like. I'll try not to pimp it too much, but when I developed it, I started thinking like from a science, how do molecules enter a root? What's the mechanism for a plant to absorb nitrogen or phosphorus or water? How does that actually physically happen? Because it kind of, I'm going to go off. I don't want to, but it
No,
stemmed
please
from
do.
people. it stemmed from like, I always thought it was a myth that, feed your plants molasses. And I always thought, man, that's a pretty big molecule. I don't know, maybe that's not what you're doing. And I always thought it was something else. So I started thinking about how do roots absorb things? And if you know, I have some questions for you. What
Sure. So it has to do with ionic bonding. So roots they emit like a negative ionic charge and they bond to positive ionic charges, and they pull it in essentially just through diffusion, and then those nutrients pass through cell walls and they get placed in the places they need to. They build proteins with amino acids and all stuff like that. So you had mentioned molasses. This is something that I actually talk about pretty often. So I don't use molasses in my teas. Molasses is a lot of people, they have success with molasses. They use it as a microbial food source. I personally am staying away from molasses. I'm staying away from monosaccharides, simple sugars as food for biology. So the reason why, so molasses will feed like a very specific spectrum of biology, and it won't feed like all life within your soil. So it'll cause like a very, very specific, set of biology to feed on it and to reproduce very quickly. It'll consume that simple sugar, and then you end up with not a full spectrum of biology being fed. You end up with just like a very small portion of your profile being fed. So I like to use polysaccharides instead of simple sugars like glucose and inside of molasses and stuff like that. The molecule itself, yeah, it's pretty big. It breaks down, it gets consumed by biology and things. Molasses is pretty rich in like fulvic acid. It's a good fulvic acid source, and it has micronutrients in it as well. The only application that I use molasses for is multiplying lactobacillus, so lactic acid bacteria serum, with molasses. This kind of helps break it down, makes it more available, and then you're not feeding your soil just glucose. Because when you pump your soil full of glucose, you end up getting bacterial imbalances. You have like a big bacterial flux. And in the soil food web, we're taught, soil food web text, we're taught to balance bacteria, protozoa, fungi, microarthropods, completely complete the circle of the soil food web to make nutrients most available as possible, and break up those locks inside your soil, so that you can really get the full potential out of your medium.
So, so you brew. So do you do anaerobic teas or aerobic teas? What do you
So
have a preference?
So I use both. They both have their time and place. I'm a big believer in everything has its time and place. Everything, all these methods, they have, they're all good for something. They all kind of lack something, and they work best when paired together. So I don't do one thing. I use both aerobic and anaerobic ferments. When setting up like a soil food web, using microscopy to check out your soil, I do aerobic teas until I see that everything is in place, everything's in balance. We have good diversity of protozoa, good diversity of different bacterias, bacillus species and things like that. And then once all that's in place and I feel like I need to feed it, I'll use anaerobic teas to feed my biology. A good thing to feed microbes are dead microbes. And anaerobic bacteria, as long as there aren't any pathogens that have brewed inside of your tea, you can introduce it to your soil and it will feed your biology. So anaerobic teas, when you look at them underneath a microscope, their bacterial density is extremely high, like much higher than what you would usually get in a aerobic tea. But you can achieve those same levels. But what you don't see is you don't see protozoa. You don't see much diversity. You just see bacteria. It's just full of not much diversity, just tons of bacteria. And so that's not really what I'm going for. In my soils, I focus for as much diversity as possible. Um, all these different species of bacteria have specific characteristics, specific enzymes that they secrete that have different properties and functions for the health of the plant, for the health of the soil, and for the uptake of nutrients. So that's my long-winded answer to your short question.
No, that's what it does is opens up a thousand questions. I should be taking notes, but I'm going to ask you questions if you don't mind.
Yeah, please do. Carry on the conversation.
So I've had it explained before, and maybe like bukashi is an anaerobic material, correct?
Mhm.
I mean, you
Yeah,
make it anaerobically.
that's a solid
And
state ferment, anaerobic
and
solid
and
state ferment.
and actually, I think there's some cool things done with bukashi. But from experience, when you added an anaerobic bukashi to an aerobic tea, something happens dramatically. I mean, it's the
Hmm.
reaction is real, and that's when you get the volcano action. And is that because what is that because that's a really good food source for the anaerobic or the aerobic bacteria, the aerobic species in your tea? Is that what's going
So
on there?
so let me ask you a few questions.
Okay,
Is there a specific bukashi you were using?
I just made it from red oats. I think it was oat bran, and I used some probably worm compost tea as a starter.
Okay.
I just made aerobic tea, and that's what I use as a starter. And I ferment. What do you do? I seal it for a couple weeks in a warm, dark place, and starts to smell like bread or beer. Beautiful smell on
Mhm.
it, and I just made my own, and that's what I'm used to.
Great. Did you use lactobacillus?
You know, I don't know. Like I said, I pretty much probably made a worm tea with the molasses.
Okay.
I probably wouldn't have added too much more than that if I recall the old days. But that's yeah. I don't know if there's lactobacillus act as I just made it strictly like I said with worm tea, AACT I think it's called.
Okay. Sorry, you broke up for just a second. I just switched my internet to Starlink, and I'm having periodic drops and stuff like that. So I apologize if I freeze for a little bit. Um, so bukashi is usually made with facultative anaerobes with lactobacillus, from what I understand. Um, I've never heard of it being made with an aerobic earthworm casting tea before. So the bokashis that I am familiar with and that I've made in the past are lactobacillus, molasses, and then a grain carbohydrate, specifically a grain. And so people will use oats and bran, different kind of meals and stuff like that. I prefer flakes and oatmeal, like some kind of flaked carbohydrate. The brands seem to grow trichoderma pretty quickly. Um, Back to your question though. So you asked adding bukashi to your tea. So it sounds like you added a carbohydrate source to your tea, which is a good practice. Elaine Ingham talks about using different grain carbohydrates as a food source for your tea, because those starches, starch is a polysaccharide. It feeds more diversity of life than a simple sugar will. And so adding a grain carbohydrate like bukashi, is on a grain carbohydrate, you're going to introduce that starch, which will feed a great diversity of life in your compost tea. And so you're giving it a really good food source. And yeah, that'll cause that life to really thrive. Um, I typically don't add bukashi to my teas, just because I make it with lactobacillus, and lactobacillus is a very opportunistic microbe. It can dominate your tea, putting it in early. So I add it. I actually don't add it to my teas at all. I top dress with bukashi, and then I'll water it in with the tea. Um, yeah, so lactobacillus usually is kind of like a post brew kind of thing. So for the bukashi, adding a starch, and you can add a starch to your tea. I do that. I add every different type of main polysaccharide to my teas. That's how I like to do it. So I'm just going to keep ranting. If I'm on a roll, I'm just going to go. Okay,
No,
that's cool.
go. I'm loving
Yeah,
this. Thank you.
yeah, totally. So I add polysaccharides to my tea. There's five main types of polysaccharides. There's polysaccharides from plants, from insects, from algae, from grain and from mushrooms. Yeah, and from mushrooms. And those are the beta glucans. And so having that diversity of polysaccharides, having the entire spectrum of polysaccharides, kind of ensures that the most biology is going to be fed as possible. And that's really what I'm like getting into right now. Like that's kind of like what I'm focusing on is playing with different levels of different types of polysaccharides and how that affects the biology in the tea to like maximize diversity. And that's my big thing that I'm playing with. I just wrote an article about that actually.
And you're affiliated with Skunk Magazine?
That was Skunk Magazine. Yeah, I wrote an
Oh,
article in Skunk Magazine. The article is called Gardens of Diversity, and it touches on using polysaccharides and how they function and stuff like that in your tea.
you know, sometimes I must stumble onto things a little bit because when I started, I sell fertilizer. I'll talk about it a little bit,
Okay.
but somebody asked, like, I always thought it was real important to have multiple. So first of all, plants and animals eat plants and animals. I came to that realization that it's a carbon cycle. So so you go anywhere in the world, right, and you have a nice meal, it's probably going to be plants and animals if it's a healthy meal, and it'll be very diverse, but you can live just fine off of it. So I always kind of took the approach to, like, I have like 20 some ingredients of meals, and everything's natural or from the earth, but I always thought, yeah, you want multiple nitrogen sources, and basically when you're using things that used to be alive, like any meal, by definition it has every element because it used to be alive. The elements don't disappear, the water evaporates. So if you have
Yeah.
alfalfa, it has zinc in it or whatever, it has to, maybe tiny quantities, but it couldn't live without it. So it's just, as
Yeah.
long as you're
Exactly.
eating things that used to be alive in a variety, that's what I think is key. And I'm glad you touched
Definitely.
upon the variety of starches. And yeah, I knew about bokashi, like oats, any kind of starch. Can you make bokashi with rice? I'm curious.
Yeah, you can make bukashi with rice, um, and that's something that um, I've actually discussed a lot about with some people in the industry. Is bukashi with rice. So I'm gonna lead into something kind of interesting here, and I hope I don't upset some people. I'm probably gonna upset some people.
That's okay. That's, you don't do anything if you don't
And
upset. So go ahead.
okay, so so Bukashi sprouts what's called Actinobacteria pseudohyphae. So the hyphae that sprouts from Bukashi isn't a real fungal hyphae. It is a bacterial hyphae that sprouts from facultative anaerobes. Um, and this is pretty much what a mycologist would call cobweb mold. But it's not. It's very different. It's very different. Because cobweb mold is, you'll see it on bread and on like old pasta and stuff like that. If you might see it like leave under your bed or something. And so the there's another practice in the organic community called IMO. I'm sure you're familiar with it.
Yes.
And so that is a grain carbohydrate that sprouts this actinobacteria pseudohyphae in this very similar way that bukashi sprouts the actinobacteria pseudohyphae. But they're supposedly completely different, as IMO, is meant to be placed on top of a mycelial layer that then creates like a mirror image. Supposedly, it like creates a copy of whatever you're putting it on top of. Um, and then it sprouts this actinobacteria pseudohyphae as an indicator that it's done, which is kind of interesting to me. Um, I'm really curious to see test results between a bukashi made from rice and an IMO collection, and really see what the difference is, because that'll be what really tells you, what you actually captured from your local area, like what you really got from.
A little buffering. While she's buffering, sometimes we use a lot of jargon, like any kind of profession, and IMO and I might be wrong, is indigenous microorganisms, if I'm not correct, if I'm right about that
That's
in
right.
the
It's
body.
indigenous microorganisms. Yeah. I'm sorry. I think I dropped for just a second.
You
Um,
did.
okay.
It's
Um,
okay. It
yeah.
happens.
Indigenous microorganisms. Yeah. It's a KNF practice. Where you undercook rice, you introduce a couple of KNF inputs like fermented plant juice, which we talk about next. I have some really cool things I like to talk about with fermented plant juice, and then an input called Oriental herbal nutrient and brown rice vinegar. So they add this is kind of like the foundation for the microbial collection and the fungal collection. But a lot of it has a lot of sugar in it, a lot of brown sugar as like the food source. I'm not super familiar with the science behind why OHN is able to collect good diversity, but they say that there's a big complex reason why OHN works really well to collect different life. I'd have to ask someone like Chris Trump or maybe Wendy Kornberg to explain that. But so anyway, um, so the bukashi made from rice would be like the rice inoculated with that lactobacillus, the lactic acid bacteria serum, and maybe some molasses or something like that. And that lactobacillus would definitely proliferate. It would multiply inside that grain carbohydrate pretty quickly and take it over. And then that's probably the majority of what you would see inside of that collection or of that bukashi is definitely going to be the lactobacillus. It's going to be the dominant microbe there. But I am curious to see what the difference would be between like an IMO collection, a successful IMO collection, and like a rice bukashi.
Well, now let's go through a little more detail. So we're going to get some rice grain, right? Some dried rice at this point, and then we're going to inoculate it with something. What? Tell me what you would inoculate it with. You just said
So
molasses.
we talking about making bukashi.
Yeah. Yes.
Yeah. So
A
you would
rice bakasi. How would you give me a recipe?
okay. So you would pretty much take 1 part lactic acid bacteria serum to 1 part molasses and then 10 parts water. And then you would use that to introduce to your rice bran. You would soak it in it. If it didn't all absorb, you could strain it out a little bit. You probably don't want to do that. You probably just want to add enough to really just get a good soak, like to field capacity, which is, you squeeze.
A little dropout again, but we'll get through this.
Sorry. Um,
Nope.
so you squeeze it until like a drop of moisture comes out, and then you would put it in like a plastic bag, like a black trash bag, and remove all the oxygen from it because it's an anaerobic ferment. You don't want oxygen in there. So you just like push all the oxygen out of this bag, tie it up real good so that there isn't any air getting in, and then as it multiplies and ferments, the bag will expand like a big balloon. And it's fine. Just let it expand, let us do a thing. You don't need to drain it out, whatever, or empty out the air, the CO2. Just let it do its thing for like 3 weeks, and then take it out, dry it, and then that's a rice bukashi.
Have you made any?
Rice bukashi?
Yeah.
No, I've never used rice. I've only used like wheat
I think we need to try. Now,
and
one thing,
oatmeal.
because you mentioned it before, I think the bran with the large surface area, the flattened out oatmeal or wheat bran is superior because of the greater surface area. Is there an analog to rice, like a rice bran or rice, maybe rice flour would to get that, because I see these little hard like little torpedoes of rice that don't have a lot of surface area, comparatively. I'm wondering, it's just a different consistency. I'm wondering if that would help.
Um, so I cut out for just a second. I didn't hear.
But you're better off.
So you were talking about using something with more surface area, right?
Yeah.
To promote aerobic conditions. kind of.
Well, no, just like when you're making bokashi, I think it's the brands work. That's all I know about is the oatmeal bran or wheat bran, where it's kind of a flat little flake. Where rice is kind of a hard little torpedo kind of shape with not a lot of surface area comparatively. So, is there an analog to bran in the rice? Is there a rice meal or a rice flour that you might try using that instead of just
Um,
rice?
I don't know. Probably. I would play with it. I encourage you to experiment.
No, it's and these are excellent things because it's so easy to experiment with.
Yeah, definitely.
If
Definitely.
and so you would use the lactobacteria.
Mhm.
Let's go with now that is that kind of one of the first steps of the ferment stage of the rice.
Yeah, so I can walk you through
Tell
how
us
to
a
make
little,
lactobacillus if you would, and
please,
lactobacillus serum. So,
please.
so you use the starch collected from rice. It doesn't have to be starch collected from rice. It just any kind of starch. I see your cat back there. And
Hmm.
so what people usually do, the normal recipe is you take some rice, you get the starch off of it by either letting it sit overnight and shaking it up, or just shake it up for a long time until you get all the starch off of it. So people might cook it a little bit to loosen up that starch. And then you strain the rice out of the liquid solution out of the water. Um, and you leave the water with the starch. It's pretty much just starchy water. Um, you put it in a jar that's 2/3 full. You cover it with a coffee filter or a paper towel, just so that bugs can't get in it. And then you seal it with just like a rubber band, whatever. So it's breathing, and you leave it out until it forms a pellicle, which is a thin little film of it's like a kind of like a biofilm, but it's a pellicle, so it's a thin little film of bacteria, of local microbes, mostly lactobacillus, just microbes that are floating in the air. And then you're introducing that. So once you have that pellicle, and you take your coffee filter off or whatever, and you have 1 part the rice wash to 10 parts milk, and going local, using like, if you can find like a local cow or a local source of milk, that's going to be best. um, but you can use anything from the grocery store, really. And you add 1 part rice wash to 10 parts milk, and then you ferment that with a breathable lid. Also, you make sure your container is 2/3 full, because as it ferments, it's going to create like a curd, like like a cheese curd. This is
Hmm.
a similar process to making cheese, but this is just kind of making cheese with like the local microbes in the air. And people will actually separate that curd and then heat it up, kind of like the same, like a mozzarella type style cooking of the cheese, and then use the cheese to eat. It's really good for you. It's good for dogs too. Dogs will love it. So once you strain the curd off, you're left with lactic acid bacteria serum, which is the whey. If people say curds and whey, they're talking about that serum, which in cheese making is usually just discarded, but it's actually a really good probiotic source for our plants. And so that serum is what would be used in making bukashi, and it's also great for a whole bunch of other stuff too. You can use it as a way to break down plant matter in your garden. You might have some like fruit or something that's going bad, that's falling from like a tree or something like that. You can throw it in a bucket with some water, and one part lactobacillus to 10 parts water, and then in a bucket full of like plant matter or fruit or stuff like that, and it will ferment it, and it'll actually harness those nutrients inside of whatever you're fermenting, and that's how you can make your own liquid nutrients at home, is with this lactobacillus. And so that's a pretty cool process too.
No, I've actually had Chris Trump on the show and
Uh-huh.
Mandy Kornberg, of course. So actually, fascinating topic is KNF. So some people are familiar with some of, but again, it takes a little while. Now going back to the cheese thing,
Mhm.
we have a local cheese maker. If I got the way you said, would that be similar? We're not using the indigenous stuff though, right? When you
You
get
could, you could totally do that. And I think that that's a good option for people who might have like a whole bunch of land, is like contacting a local cheesemaker, so that they can get like a bulk amount of it, but I definitely encourage people to make their own from their own garden, collect the side in your garden in like a shady area, because then you have microbes that are adapted to your local climate, to your specific microclimate, into your garden. It's always going to be best to use microbial inoculants that are made in the area that you're using them in the microclimate that you're using them in, closest to the garden as possible. That goes for everything. That goes for compost, earthworm castings, insect grass. You and even when you're making KNF inputs like fermented plant juice and stuff like that, you want to make them with plants from your local environment, because all of the microbes on that plant matter have adapted to that local environment, and are more hardy. They're able to survive that climate, and multiply faster. You end up with healthier soil, healthier plants by using local inputs. So making lactobacillus locally in your garden is going to definitely be better. But yeah, if you have access to like a cheesemaker and you have a big farm, yeah, go for it. That would totally work too.
So like fermented plant juice, you hear a lot about papaya and such, but because Chris
Mhm.
Trump's in Hawaii, where it's local, and you probably can find some natural, not from the supermarket. Where I live, there's tons of berries, wild berries, but that would probably be a real good source because they're not been in the store,
Yeah.
they haven't been touched.
So let me
Yes,
let you in on a little trick here. So
please.
berries are one of my favorite, if not my favorite, input to ferment for fermented plant juice. Um, and one of the reasons is that they're premature berry. The premature berry is really rich in a hormone called jasminic acid, which is
Hmm.
a stress response hormone, um, that will it just causes massive growth in your plants and like huge vigor. So you take berries that are still kind of green, that are just now starting to change color, and you harvest them. You harvest
Hmm.
as many as you can, and you take equal weight your berry to brown sugar, and you stomp on it, or mix it up, crush it with whatever means that you can, to try to get some of the juices out of it. And then when you mix it with the brown sugar has a high osmotic pressure. So fermented plant juice is an osmotic extraction. Um, it's also a fermentation, but it's mostly as an osmotic extraction. Um, so you mix it, weight of berry to weight of brown sugar, and you weigh it, between 3 and 7 days, depending on how warm it is, and it will extract the juices from that berry. And the premature ones are super rich in this jasminic acid, which will raise your plant bricks. It does all sorts of great things. But so like when a plant is attacked, like by bugs or by like an animal, usually most like an animal, something eating it, um, it has these stress response hormones, and
Hmm.
jasminic acid is one of them. And it signals the plant like you're gonna die. You need to hit the survival button, kind of. And so it releases this jasminic acid throughout the vascular system of the plant, um, and it produces it. It catalyzes it, and catalyzes its production, and causes the plant to just have like immediate growth response. And you see like new growth and stretching, from this stress response. But we can harness that jasminic acid, and introduce it to the plant without having to stress it out. So we get the same effects of that boost. We get that big boost by using fermented plant juices from different inputs that are rich in different hormones, organic hormones. So the berries are great. I'm glad that you brought that up, because that's one of my favorite. Um, Another really good one is bamboo shoots, which are pretty
Hmm.
hard to get a hold of. But if you can find like fresh, soft bamboo shoots,
Hmm.
that bamboo is one of the, I think it's actually the fastest growing plant on the planet. And you're essentially harnessing that essence. You're harnessing that plant essence, and then you're introducing it to your other plants. One thing about using fermented plant juices, is that you're supposed to use them in conjunction with the full KNF regimen, with your microbe source, which is lactobacillus and indigenous microorganisms, the IMO. This is all KNF system stuff. And then your OHN, your brown rice vinegar. Then there's a few other, but those are the main ones. So you have your microbe source, you have your enzyme source, which is your brown rice vinegar, and then you have your lactobacillus, which acts as an antipathogen. It's also another microbe source. And then you have your source of hormones, which is the fermented plant juice. The fermented plant juice isn't very good at available nutrients. They don't have much nutrients in it because they haven't really been broken down. They haven't really gone through like the chelation process. So they need these other elements to really kind of make them available. Oh, and fish amino acid, I'm sorry. The amino acids is very important also. And so these are all like the chelating compounds. You know, we have our amino acids, our enzymes, and our microbes. And that's how pretty much everything is made readily available in the soil is by these compounds or these, compounds. So you add you add your FPJ to these different compounds and it unlocks them. So you never want to use an FPJ by itself because it's a lot of sugar. It's made with brown sugar. You don't want to just introduce a whole bunch of simple sugars, monosaccharides to your soil because it causes a bunch of bacterial imbalances. So you add it with all this other stuff and it helps break it down. It helps it be consumed quicker so that, things in your soil can't get out of whack and stuff like that.
I got a couple questions for you.
Yeah, let's hear them.
I'm going to start off. You got some really good words, some big words.
Okay.
Or is your background in actually in science, or is this just from being because I know you can learn about anything if you're interested in it, and it's a cool part about learning. And yeah, what's your background? Because tell me that first, and I got some more questions.
So no, I don't have a formal education in any sciences. Actually, I have a degree in business management and social and behavioral sciences. Oh wait, so I guess it is kind of science, but like
not really.
Not really. No, it's cool. No, I feel the same way. So I was growing cannabis to put myself through college, and then I never stopped. And I just found such passion in just things that help the planet, that help our body, that grow nutrient dense foods. I just found like a real passion in the science and all this stuff and just dove into it, head on. And I'm kind of like a habitual rabbit holer where I find like a rabbit hole of information and I just dive into it. And I kind of like obsess over it almost and like learn as much about it as I can until like I feel like my information intake is kind of plateaued and then I move on to something else. And yeah, so I'm big into microscopy. That's kind of like an endless rabbit hole that doesn't ever really stop. You never learn everything about microscopy. And then, I'm working on soil science stuff, which is also, you can never really learn everything about a science that, we don't know everything about. It's like studying astronomy. So no, I don't have a background in sciences.
Well, you're very well versed, and that's awesome. I have some questions, and because you got me thinking about a few things. When you talk, I think it was jasminic acid is a hormone that's produced by newer fruit. You were saying, you want to use it like when it's before it's ripened. Was my take on what you were saying.
So premature fruits, not all of them. Berries are a very high concentration of jasminic acid. Premature apples have a very high concentration of jasminic acid. Bamboo, which I had mentioned, also has high levels of jasminic acid. And then jasmine flowers have a high level of jasminic acid.
That will kind of make sense.
Yeah, totally. And so, yeah, exactly. And so it's a response to damaged tissue. So you can get jasminic acid from anything that's been like damaged. But I mean, I would definitely get it from one of the 4 inputs that I mentioned, or just like do some research on the local plants in your area, the invasive plants in your area. I recommend this to a lot of people. is kind of take an inventory of what you have growing on your land that you're farming with, and research every single type of plant that you have. Like identify what it is that's growing on your farm, and see what like the profile is of what's growing there. Um, and then you can ferment those inputs for different growth stages
And then,
for
yeah,
your
that's kind
plant.
of what is I actually have a ton of bamboo. I love bamboo. I got all kinds of
Okay.
bamboo. And you were talking about the jasminic acid produces that real growth spurt, right? Am I
That's
right
right.
about that?
That's right.
So
Yeah.
so is there any logic involved, like say as a fruit is ripening or it's ripe, are there any other hormones that are secreted that maybe you want to use fruit later,
Yeah.
fermented fruit juice like later in your cannabis cycle life? Because if you're seeing that vigorous growth, maybe that's a good time to do it in the veg stage, or right or when it flips to flower. You know.
Absolutely. The so you want to find something vigorous, like a vigorous vegetative growth,
Hmm.
like bamboo, or like a stinging nettle, or some kind of invasive weed that grows really quickly that you have on your land is going to be good. And then that's going to be good for your veg growth. Transition into flower, I use the premature fermented berries, like right around week 3 when you start seeing like pistil growth really starting to throw.
Hmm.
I'll use fermented plant juice um, from like berries, from premature stuff like premature apples and things like that, and then I'll use that up until like week 5. Not too much though, because if you use plant growth hormones too often, your plant will get used to them and stop responding to them. So week 3 to 5 I'll use premature fruits, and then it's great because once you get later on in the season, then you have more mature fruits that then you can
No.
use for your more mature plants. um, and so the hormones change, um,
Yes, they do.
and yeah, exactly. And then you yeah, so then you use like more mature fruits, more ripe fruits during the ripening stage and stuff like that.
So
So
yeah, so you ferment kind of like in parallel with your plant growth. That makes a lot of sense. That's so
yeah,
cool.
yeah, definitely. So the earth kind of gives you exactly what you need when you need it.
Mm-hmm.
Mhm.
Mm-hmm. Great.
Yeah.
Where else you want where else would we want to go?
Um,
I'm gonna go way back just for a second to when we talked initially about like the mechanisms for say water or whatever to go inside a root, and you mentioned ions, which I come from a real basic science kind of thing where the only way something in nature will move is if it's pushed or pulled. That's it. That's the only way things can be moved. It's with Newton's laws, right? But
mhm.
salt-based fertilizers, when you mix a salt and water, you get ions, right? Natural, so they're charged little particles. So they're kind of like an IV to a plant. It's that charge is there, and the plant's looking for different charges. And I think it's from what I've heard is there's little proteins on the root hairs or whatever that rotate out like in nature a plant it doesn't go to another field where there's more phosphorus when it's fruiting. It takes phosphorus when it needs it. It takes nitrogen when it needs it. And I think it's those little proteins that rotate out and grab a phosphorus or a nitrogen or whatever, and then rotate back in. I could be wrong, but that's kind of how the nature does it, and I'm not real sure about that. But
So
help me.
the rotating proteins. I'm not super familiar with that. I imagine it's enzymes, proteins, um, different negative ions that match like a positive ion that then bonds to it and pulls it in. I believe a lot of that is just diffusion. The plant will
Yes, yes.
transpire the
To lower
water out of
to
the
lower
the leaf surface.
concentration.
Hmm. So it transpires, and then as it transpires, it creates a um, I guess it would be a high osmotic pressure, yeah, in the root system, and then that diffusion would pull water in, and then the different um, I don't know if it's proteins. You might be right though. Enzymes or proteins that will then bond to um, the ions, the dissolved solids, the electrolytes in your water. so the elements, they are dissolved into water. They alter the electrical conductivity, as ions, and then that's what your ppms are, or your EC is the how many dissolved solids, how many ions, or the concentration of ions in your water solution, which are just the elements, the readily available elements,
Okay, because I had a discussion with somebody
nutrients.
about feeding plants neem meal, and
Uh-huh.
someone had a concern about, well, some of those, because neem meal does have different um, proteins that are poisonous, right, to different things. That's one of the reasons it's an antiseptic or whatever. But
Mhm.
would it be would it be correct to say that a molecule has to be water soluble before it can go into a plant, be absorbed by the root? It doesn't have to be
Not
dissolved in water, not
necessarily.
necessarily.
Not necessarily. So there's, I mean, most of the time, yeah. So there's enzymes that allow full proteins to be absorbed by plants, without them completely breaking down. Proteolytic enzymes, and that's so like yes and no. There's always exceptions. Sorry, but so no, I wouldn't say necessarily they have to be completely water soluble. Um, but I would say in most cases, like yeah. Um, I'm sorry, I don't know much more about that.
No, it just got me thinking because some of those poisons are not water, like terpenes are not water soluble. So I don't know,
Mhm.
feeding terpenes into the soil, in my opinion, won't do anything, because you can get terpenes, are readily available now, synthetic especially,
Feeding
but
terpenes into the soil.
they wouldn't. I don't think, because they're not water soluble.
No, I wouldn't do that.
No.
So you definitely don't want to add oils to your soil. That causes all sorts of problems and stuff. Um,
Yeah, and actually terpenes, pure terpenes are they can be quite caustic.
yeah, that would cause a lot of damage. Um, so no, I definitely wouldn't do that. If you're trying to increase, like terpenes, there's a lot of different microbes that are I'm sorry, there are a lot of terpenes that are only created by specific microbes. So, this is one of the reasons why we see that uh, organic growing, living soil grown cannabis has more terpene content, is because of the microbe diversity. And a way to attract microbe diversity is diversity of enzymes also. And so one of the things that I practice and a lot of people practice is diversity within your cover crop. So creating diversity of enzymes inside of your rhizosphere.
Hmm.
Um, so this is kind of anecdotal. I don't know if this is necessarily true, but I heard it recently and I found it to be fascinating. So people are saying, I've heard recently, that adding different plants high in terpenes like lavender or like herbs, that they really smell like really strong, have specific terpene profiles, that their enzymatic root exudes or exudates in their rhizosphere attract specific microbes that create those terpenes, and that if you introduce them to your root zone, to your rhizosphere with your plant, you're attracting those same specific microbes with those enzymatic root exudes that then create those terpenes that can be absorbed. So the terpenes aren't made in the rhizosphere, but the microbes help the plant express those terpenes. So you're introducing the plant with a specific terpene profile that secretes enzymes that attract microbes that create terpenes. um, does that make sense? Did I explain that well?
Yeah, well, it's all kind of complicated, to be honest
Yeah.
with you. I had a little different understanding of terpenes.
Okay.
Can I give you my and steer me where I'm wrong? But it starts with photosynthesis, right? Water, sunlight, and carbon dioxide, and chlorophyll makes a simple sugar, right? And that consists of carbon, hydrogen, and oxygen, which all comes from either water or CO2. So, and terpenes are made of three elements: carbon, hydrogen, and oxygen. THC is made of three elements, carbohydrate, so is CBD. So are a lot of things are just those three elements, but arranged differently. So sugar is one of the first carbohydrates that, well, it's the first one, and then there's mechanisms in the plant that fold it and rotate that carbon, hydrogen, and oxygen, and then when you get five carbons, you get your first terpenes. And it's interesting. I have no idea why, but it's in units of five. It's 5 carbons and 10 carbons and 15 carbons, and then they're too heavy to be volatile after that, I think. So it folds and becomes THC or CBD, but it's all originally from the carbon dioxide and water. That's where
Okay.
it's I look at this plants as I have this crazy idea. The plant's a machine, and there's machinery like ATP and chlorophyll. Those are the machinery of the plant, and the plant takes CO2 and water, and that machinery turns it into things that we like. Especially psychoactive plants are always, there are 3 or 4 elements: carbon, hydrogen, oxygen, and sometimes nitrogen. And those all, interesting enough, come from the atmosphere, which it doesn't come from the soil. I always thought the terpenes are from the atmosphere because there are only 3 elements, and I don't know, maybe I'm completely off on that, but I always thought it came from the carbon dioxide, not not an enzyme or something. Help me out
So,
here.
so yes, yeah, you're right. That is on point. That is correct. Um, so these processes that you're referring to require enzymatic biocatalyzation.
Okay.
So like the process of absorbing carbon requires an enzyme that delivers that carbon. I forget what it's called. It starts with an R, rubas something. And then
It eludes me at the moment too.
yeah, you know what I'm talking about.
No, I don't.
Oh, okay. Um, so enzymes play a huge role in all of this. And I wasn't trying to say that the microbes in the soil are creating the terpene. It's the enzymes and the relationship to the microbes um, that give the roots what they, oh fucking A. How, maybe I'm not explaining it. Maybe I don't fully understand it. Because like I said, this is kind of anecdotal, and this is something that I've heard. But I do know that specific microbes, when present in soil, are responsible for specific terpenes to be produced, and that's not anecdotal. That is confirmed science. That's published in scientific articles, by universities and stuff like that. That specific terpenes are only made by with the presence of specific microbes in the soil. Um, I don't understand everything.
Oh
Um,
no, I think it's an amazingly and fantastically sometimes complicated, and when I started doing, like I said, when I started looking, say, okay, how does phosphorus go into a root? What's the mechanism? What's going on there? And it turns out, it's not that well understood, and there's some, especially with mycorrhizae and the living soil, and actually it's starting to. I think Rutgers University, I've gotten some really interesting papers from them where they're, because the significance of this is huge. It's food, which is really high on the pyramid of what we need, and being
mhm,
efficient creating food, and that's what's so cool about being in this community, is that we're doing that. What you're talking about, making things sustainable, and the idea of bomb making materials to plant our, to sterilize the soil and make monocrops. We're learning, we're advancing it past that, and universities are starting to do some, just, it's really new, the research they're doing with, and it's not that well understood. It honestly isn't, not what I can find out.
mhm.
And that's what's fascinating. There's the unanswered questions to me.
Definitely. I love that we're kind of moving into this new, it's almost like new technology that's really been around since like the
Yes.
beginning of time, you know?
No, it's like
Um,
like a tea to me is an incubator for the goods, for many whatever you put in there. Good in, good out, bad in, bad out. But you're just making, taking it, you're just tricking multiple. You're using math. You're multiplying, or
yeah, definitely.
exponentially, and you're basically taking what nature has and just kind of manipulating a little bit. And isn't it a cool profession to be in? The people we get to hang out with. Come on now,
Yeah, definitely. Definitely
it's a
very cool people.
very interesting people too.
Yeah, yeah, absolutely. I think we all have different things to contribute. We're all very different, and we can learn a lot from each other. And sometimes some people are going to come up with something that you know other people aren't going to come up with. So,
No, that's why I love to talk with people on the show. I used to be also an inspector for a certification program, so I got to see a lot of farms, which it's just the more you see and the more you absorb and the more open mind. I learned a long time ago. Not it's hard for me to say I've gotten burned. You can't do that or you can't do this. I got to be careful there because I'm not real sure. I know some things work from my experience, and that's all. That's kind of all we can go on, what our road was. And
what do you
I'm,
wait? What are you talking about? Can't use what you're talking about, like IPM
wait,
stuff? What
I
are you
don't.
talking
I forgot what I just said.
about? It's okay. It's cool.
I'm not.
We can just move on.
That's how in the moment I am. Is I don't remember the ones I just talked about.
Oh, that's
But,
good. That's
but
cool. Yeah.
are you in Humboldt, by the way?
No, so I'm in Southern Oregon right now.
Oh,
Um,
okay.
yeah, but I grew up in Northern California.
Okay.
Yeah.
Uh, yeah, I did a lot of farms in Southern Oregon. I'm in Crescent City,
Oh, cool.
so you come here when you want to get cool in the summer.
Nice. Yeah, definitely. Yeah, I'm a little
Applegate
past
Valley, by any chance?
no, Rogue Valley.
Oh, okay, okay, a little
Yeah,
farther north.
yeah, yeah.
Oh yeah,
Yep, yep.
yeah, beautiful. A lot of farms in Southern Oregon.
Yeah,
OMG.
yeah, there's a lot of growers, a lot of good growers out here for sure. It's good competition.
Speaking of a
So,
competition, did you hear? I did a Jefferson Cup.
mhm.
I think it's so cool. Did you ever hear about the Jefferson Cup here in Crescent City?
No, I'm not familiar with it. I just moved up here a year ago.
Okay, I
Yeah.
want to brag a little. I had a can, like a show like the Emerald Cup. I did it here in Crescent City, and I broke some rules. We had Southern Oregon versus Humboldt, so
Okay.
we crossed
Some
state
rivalry.
lines. So I had a two-state show, and I'm really proud of it. We pulled it off. It was kind of a cool show. 2018, I think it was.
Nice.
But no, that would Crescent
Who won?
City's actually a guy. First and second place was Humboldt, and then a farm in Southern Oregon. So they were very close. It gets
Okay.
how can you know what I'm saying? It
Yeah, for
gets
sure.
pretty subjective when it gets
Yeah.
on top shelves. But it was cool because here in Crescent City, 100 mile or 60 miles west, east, 60 miles south is some of the biggest growing areas in the world, and I took advantage of that. But it's a really
Yeah.
cool area. I love it. Yeah.
Yeah, that's nice.
Well, enough
That's cool.
about me. So you're in Southern Oregon and work for Skunk Magazine,
Hmm.
and I was introduced to you through Wendy. How do you work with Wendy?
Oh, I've never worked with Wendy.
Oh, you
Um,
haven't.
she's just a friend of mine through the internet. I've never actually met her in person. I drove past her farm like a few weeks ago and tried to hook up with her, but that it didn't happen. So, I guess just through like the KNF community. Um,
Okay.
yeah,
Interesting people.
definitely.
And you mentioned Elaine Ingram. She's one of the gurus, I can say. She wrote some of the real interesting books. I've
That's
heard
right.
I've heard different things. I've never been to one of her lectures. It's something on my list to do. But
So she is definitely a powerhouse of information. That one. Um, and she writes curriculum for I think Oregon State.
oh really?
Maybe I'm wrong. She writes curriculum for some colleges. Um, but yeah, she puts out all the soil food web information. She has a lot of microscopy expertise. She's a doctorate in horticulture. She's definitely one of the people up there that should be looked up to. She knows her stuff.
No, I think she's up there. Jeff Lowenfield has done some interesting things. Chris
Yeah,
Trump, yourself, and Wendy, and all the people who are kind of taking this and putting their own little spin on it and looking at it differently, which is how you have growth. Pun intended.
totally,
Pun,
yeah,
pun
absolutely.
intended.
You know, yeah, everyone has their kind of ideas that they subscribe to. They're a little bit different than the next person. I know that Elaine Ingham, she has a very specific ideology that she follows. Same with Chris Trump. Well, I don't want to speak for these people. I'm sorry, but they Yeah, everyone contributes different ideas. And I have some ideas that are a little bit different than a lot of the communities, the KNF community. I have some different ideas. I don't use strictly KNF all the time. I use a lot of their inputs, but I use them in a way that isn't the KNF process. A lot, like instead of using indigenous microorganisms, instead of using IMO, I use compost tea as my microbial inoculant. And instead of using fish amino acid, I use aloe vera. Um, and that's not any aversion to meat products, although that is a positive. It's more of that aloe vera has 19 of the 20 different amino acids. And then, amino acids are what create proteins. And so if there's a different type of protein that needs to be created, there's a different profile of amino acids that need to be uptaken and used, or, as bonding purposes and stuff like that. So for sake of diversity, I like to use aloe vera as my amino acid source instead of fish amino acid. But that product also has its own benefits, or that input. Using fats, adding fats to your soil has a good, it changes the consistency of your soil. It'll help with consistent microbial growth and has other properties that it brings and stuff like that. And so it has this time and place. But so, I like to change things around a little bit. I do things just a little bit differently than some people. And, I'm super down to spread that information however I can. And if you like it and you want to subscribe to it, that's great, and if you don't, that's fine. I'll just do my thing and you do your thing, and we're both right, and no one's wrong, so
That's right. And
yeah.
and like you were, yeah, I'm glad we talked about berries because a friend of mine introduced me. He goes, "Ben, you gotta see this guy Chris Trump. He's doing some real interesting things." And he started fermenting his fruit juices and plant juices, but he went way out of his way because he saw papaya, so he thought he had to have papaya. And by the time he got it from the grocery store, it probably wasn't the ideal source for your juices. And you get on tangent. Well, that's what he does, or that's what she does. So I have to do papaya, but you really don't. You do what's local. I think, and experiment a little bit. Go outside the box, and
Absolutely.
it's
So
so
that's one of the
key.
reasons why, if you follow me online, I don't usually talk about the specific inputs that I use, because I don't want people to go to the store and buy them.
Exactly.
And I did tell you that I use aloe vera and berries, but
I grow
it's
my own,
definitely, yeah. Yeah, berries are, yeah, pretty much
and
everywhere.
aloe. I grow a lot of that too.
Oh, great. Yeah, you should try
I've
it.
never I've never thought I've tried spouting seeds in it, which works pretty good. But
Yeah, it does.
no, I'm gonna try that. In fact, I grow a lot of things I can't always talk about, but
Oh,
I love to grow
okay.
plants.
Uh,
I love I grow a lot of plants.
yeah, so God, we just kind of got off tangent. I forget what I was talking about. But yeah, the aloe vera, you can use like a quarter cup per gallon of the juice of aloe vera.
Hmm.
Um, a lot of people like to scrape out like the gel on the inside and not use the rind. Um, I just blend it all up together, um, and then
Yeah.
just use it like that, because there's a lot of nutrients in the rind and stuff, and as long as you're not adding a whole bunch of way too much non-composted fresh green organic matter to the top of your plant, it shouldn't really be an issue. Um, but yeah, yeah, it's
No, and,
also.
and you're also going to get the benefit of what's lands, the natural bacterias and fungus that just lands naturally on the outside of the plant, which is probably not real, which is going to help break it down eventually, right?
Yeah, absolutely, absolutely. Also, aloe vera is a good source of B vitamins, which usually only come from bacteria. Bacteria vitamins. I think that's why they call it that. Um, usually you get them from meats and stuff. Uh, but so
You're breaking down again. My
okay.
grandma taught me about aloe vera, putting on your, like a burn or a cut or something. It really seems to make a quick difference in healing too. That's how I got introduced to aloe vera.
So that's the salicylic acid, which is also another stress response hormone. It's really good at boosting systemic auto resistance in your plants, so the immune system response in your plants also. Um, sometimes people, if they have like a russet mite infestation and their plants are just struggling super bad, they'll add salicylic acid as a way to help your plant recover. So aloe vera is a good source of that naturally, that you can use to give your plants a boost. So
See,
so
cloning gel. You got me thinking. Cloning gel.
cloning gel, yeah, people use aloe for cloning gel,
Never
definitely.
tried that.
Yeah,
Never tried
I
that.
I'll do that. So I do, if you want to talk about cloning, I like to do
Yes.
like a KNF kind of approach to cloning. There's a solution in KNF called seed soak solution, I think is what it is. It's SES, but I think they call it seed soak solution. And it's the OHN, the brown rice vinegar, and an FPJ. Um, and so I'll do that, and I'll soak cocoa core with it, like a really fine cocoa core,
Hmm.
and then get it to field capacity. And then I'll use, I'll take my cuts from my clones off my mother's, and then I'll dip them in the aloe vera, let them sit there for a little bit, depending on how much time I have, and then put them in the cocoa core.
Okay,
Um,
a little bit is a little relative. We're talking 30 seconds or 20 minutes or what? What?
I mean, it really depends on how much time you have. Sometimes you're making a lot of clones and you don't want to sit there for 5 minutes each time you're making a clone. So I mean, you can just dip it and use it to seal up the wound. Use the gel to kind
Okay.
of close that wound and then to stick it right in there too if you want. But definitely leaving it in there for longer, kind of, or
Sips. Good to see you. We're having a little buffering issue. Hey, thanks for tuning in, guys. It looks like a pretty good show. I don't know. I'm fascinated. I'm enjoying this topic. There's a lot cool stuff. Berries, aloe vera, bamboo, bam. I got a lot of good tips tonight. You're back.
I'm back. Sorry. So yeah.
Okay.
So about the using aloe as cloning gels. So yeah, people say that the longer you leave it in your aloe, the more compounds will be absorbed into your cut, and then the more success rate that it has. I don't know if that's actually true. There's so many things that are just said that I don't think are ever actually tested, and maybe people have success for whatever reason, and that correlation may not be the causation or whatever, but
No, that's really common
uh, yeah. So
for sure.
so I'm not gonna say like leave them in for as long as possible, because I've also seen that using too much aloe vera will actually break down. The enzymes can be so strong that it'll actually melt the proteins, and cause like plant damage. And that's something that I would like to mention real quick, about using fermented plant juices using FPJs. Is that there are some inputs that have incredibly strong enzymes that if you use them, that you can actually damage your plants. And so, like always, when you're trying something new, just use it on like a small portion of your garden. Don't do the whole thing when you're doing something new. So specifically, pineapple. And this is a mistake that I made, unfortunately. I don't even know, maybe 4 or 5 years ago, um, where there was a talk of the concept of you use big fruits have big fruiting hormones, have a lot of fruiting hormones. Big roots have a lot of rooting hormones, was kind of what was being discussed. And so immediately what came to my head was pineapple. Like, pineapple is huge, it must have a whole bunch of rooting hormones in it. And I don't really know if that's actually true either. If a big fruit has big fruiting hormones, or if a big root has big rooting hormones. It's just something that was said on, and online, and people pick up these ideas and run with them sometimes. But so I fermented. I made an FPA out of pineapple, and I gave it to my plants. And at the time, I did not know about. I think it's pronounced bromelain. Bromelain, which is a proteolytic enzyme. It breaks down proteins. And I melted my roots. I fucking killed my plants. Complete crop failure. Fucked my whole fucking ground up. It was tragic, and it killed all the worms. It destroyed my grow. It destroyed it overnight.
Inter-
It was
Interesting.
it was tragic. It was so tragic. Um, so just stay away from
So,
pineapple FPJs. Don't do that.
so can I make a generality and say enzymes are usually they're a molecule that breaks down another molecule?
That is one of the functions. So I mean, enzymes are biocatalysts for a lot of different processes. Like the absorption of gases. The they attract different microbes. They do a lot. They do a lot of different things. Enzymes are super super fascinating, um, and that's another kind of rabbit hole that involves a lot of exploring um, enzymes. They're big
I'm
big science.
I love enzymes. In fact, when I like I said when I made my tea, I looked at, well, how do plants absorb things?
Hmm.
Number one, you want a lot of surface area, right? You want white fluffy roots with a lot of white hairs. That's where it gets its stuff. In the mycorrhizae actually extend that, right? The hyphae actually extend the root zones, from what
Yep.
I understand.
Yeah.
But I use an enzyme called cellulase because a root is cellulose, pretty much, right? The
Yep.
outside of
Yep.
a root.
Hmm.
So I guess because I like to strip that little bit of out later, strip a little bit off, which I think it must be doing. But when you're talking about melting roots, I guess you want to be careful not to
Use
amp,
too much.
use too much cellulase because it breaks down the cellulose a little bit. But so far it's
Definitely.
in small quantities. I kind of I kind of like it. It's
So
kind of like
that
a burn wound. They scrape it off to make it heal, kind
uh-huh.
of deal. I think
Yeah.
that's where I got that idea.
I mean, that's great. What that would help with is breaking down your compost. So
Yes,
cellulose is actually is a polysaccharide, um, and that's it feeds biology. It's a complex carbohydrate.
it's hard to
Um,
break down though for a lot of biology, right? Or at least
it,
mammals. Most mammals or cows do it.
yeah, yeah. So that cellulase. Is that how you pronounce it?
Cellulase, I don't know. I
Cellulase.
would.
That'll help break it down. Just like chitinase will help break down
Hmm.
chitin or chitin or however you pronounce that word. I read a lot of these words and I don't know how they're pronounced. Um,
Yes, I use glucanase also, by the way, which breaks down glucose. And I don't know if it helps. Another thing about enzymes too, which I learned from my partner who has far superior knowledge than I have, is that enzymes typically work in a real narrow pH range or temperature range, or that's why we need it's real important that our body regulates temperature because enzymes are pretty finicky molecules in order for them to work. Like the pH has to be right, or there's different levels of how well they work. There's a lot of factors with enzymes, right?
so I caught the last like 10 seconds of what you just said.
Oh, you're kidding, right?
I'm sorry.
No,
So I heard you say that pH will affect the function of enzymes.
yeah, because when I was doing my tea, I got a enzyme that broke down some I forgot what protein it was, but my partner goes, yeah, we could use it, but it works at a pH of 1. He's very knowledgeable in the business. So I said, well, I guess that's not very good for us to use. Then it's not. He goes, no, it's not really doing anything until it's pH of 1. So I think sometimes, like in our body, some of the enzymes, it's real critical for certain temperatures, right? And I imagine pH is critical for some enzymes to work correctly. I'm guessing, maybe.
Oh, I don't know about
Anyways,
that. Um, so I mean, I do know that there's a specific range where nutrients are readily available, and because of pH, because of the potential hydrogen within the uh, solution in your soil of your moisture.
and I think living soils, nature buffers, like our body buffers pH to make us. It has that just mechanisms to buffer pH, and the same thing in living soil and plants. When you're doing salt-based, pH is so critical. I don't think it's so much critical in plants. Not enough to change it too much. I think nature buffers itself right around 7 pretty well.
Right.
Yeah.
I never pH my water.
Or I don't flush either. I don't know. I always thought that's a myth, but maybe I'm
Yeah,
wrong.
no, I don't do that either. That doesn't make any sense to me. That's old hydroponic knowledge that somehow got passed into organic world, and people have these ideas of what they think it needs to be done to grow cannabis because that's what they were told in the hydro store, which was the only place to get information for some people back in the day, you
No,
know?
we
And
had a real interesting industry how that worked, and when it was very lucrative. I mean, there were years probably when you were in college, it was pretty lucrative. It was when I was. But and then you get away with selling all the crazy stuff, and yeah, I think going back, kind of going full circle and using, yeah, I haven't measured pH or ECM or anything in years, and
you
there's
know what
no
I
real.
you know what I do monitor
What do
is
you monitor?
dissolved oxygen levels.
Oh, okay. How do you
Yeah,
do that? How do you
with
monitor
a
that?
with a dissolved oxygen meter, of course.
Of course.
um, so there's a big part of microscopy and making compost teas and stuff like that is identifying anaerobic conditions in your compost teas.
I want to
Um,
get into this too. Thank you.
yeah, totally. I'm glad that we're touching on this.
I'm
Um,
getting into this now.
so um, so one of the methodologies that are used. Oh damn! Did I drop again? No. Okay. So one of the methodologies used to determine if a tea is gone anaerobic is the density of protozoa, specifically ciliates, right? So protozoa are a key role. They have a key role in making nutrients available. So the bacteria will break down a nutrient, then the protozoa will consume the bacteria, and then its excrement, if you can even call it that, is you know, it's how nutrients become readily available in the soil food web, right? And then microarthropods will eat the protozoa. But back to what I was saying, the ciliates, there's this kind of common misconception that ciliates are signs of anaerobic conditions, because if you see a bunch of ciliates, it's only because the tea went anaerobic. And so there's a little bit of misinformation here that the idea is kind of on point. So ciliates are aerobic protozoa. Protozoa are aerobic, but they consume oxygen. And so when you have a high density of these protozoa, of these ciliates, they consume all the oxygen in the solution, and it creates anaerobic conditions. And then those anaerobic conditions will they provide an environment for anaerobic pathogens, anaerobic fungal and bacterial pathogens, which can infect your plant and can infect you, and especially in people with compromised immune systems and stuff like that. So in microscopy, we monitor a compost tea, or even just your soil, for the ciliate count and for the protozoa count. And you want a diversity of protozoa.
I have more questions. Come on back, Luna. Come on back. We'll get her back. Got you
Sorry,
back. That's all right.
sorry.
So,
The,
quick
the
question: is trichoderma what would that be? A protozoa or a ciliate or something else?
trichoderma is trichoderma. I think it's a technically it's a single cell fungi. I think. I don't know. We could Google it really quick. I'm not sure exactly, but I'm pretty sure it's a single cell fungi.
I've never been accused of that.
Accused of what? Oh,
Being a fungi.
okay.
But
Nice,
anyways,
very nice.
I don't
Good.
know.
It took me
So
a
yeah,
second.
I like trichoderma myself. I heard it helps like with fusarium. I read somewhere, so it's a Battles fusarium. Could be
So
wrong.
it from what I understand is it outcompetes fusarium. It'll spread throughout the soil and pretty much competitively exclude the fusarium and a
It's
lot of other things
voracious.
also.
Yeah, it's very
Yes,
voracious. That's why sometimes they
exactly.
mycorrhizas label no trichoderma, but I add it, to be honest with you.
So
I add
because
it.
it can outcompete the mycorrhizae. Um, but
Completely,
if you
you think, or just you know what I'm saying? It's like you wouldn't use that. You wouldn't use trichoderma to sterilize water or anything. You
oh,
know what I
excuse
mean?
me. No,
So
no way. No, no, no. So it needs to have a food source and it needs to spread. So completely, people say it does.
yeah,
I've never explored every single square nano inch nanometer of a root zone to look for mycorrhizal colonization versus, trichoderma colonization, and it would require pretty extensive. But people say that, yeah, it completely outcompetes it.
Okay.
And again, I don't know that. And I feel like that's one of those things, like, how would you know that or not unless you actually scoured every single portion of your root zone with a microscope, you know?
Okay, so
So,
let's talk about the microscope a little bit because the things we're talking about, the difference between bacteria and say, as I think a ciliate, is huge in magnitude, correct? As far as how big they are.
yeah,
Um,
definitely.
how I've looked into it a little bit, and actually using a microscope is a bit of an art. It's not something you just put on a slide. There's a lot of technique, and it's a skill for sure to use a microscope. How do you measure bacteria? Isn't it like stained, or there's a process where you can't look at it directly, or am I wrong about that?
So when you make your slide, you put a small drop on a slide, you cover it with your slide
Okay,
cover. You close your diaphragm completely. And that's
what's that?
it's a little thingy. It's like it almost mimics like your eye. Your eye like opens and
Oh,
closes
okay.
to
Oh,
like
yeah.
filter out
Okay, I
light.
know what you're saying. From the light source.
So this functions like that. So it constricts, you constrict the light source to be
Yes,
smaller.
it's like
Um,
a shutter in a camera, or what's that? An aperture.
right.
Yeah,
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 like that.
I got
I'm
it.
not super
Okay.
familiar with cameras, but so yeah, it'll close the light. It'll it limits it. It limits the light exposure. And so, because if you have too bright of light, um, the microbes are completely transparent. You can't see them. So
Okay.
you have to close your diaphragm completely, right? And then you'll dim your LED or your bulb or whatever it is, until you can see it properly. I start at 40x, and then you do most of your observation at 400 times. Um, yeah, and then sometimes you might go to 1,000, but that requires like making an oil,
Mm-hmm.
using oil, which you pretty much just drop a little drop of oil on top of your slide, and then you slowly put your lens right into it, and then once it touches it, you pull it back just a little bit, and you'll see that the oil will kind of create like a hourglass shape, like an
Mm-hmm.
hourglass kind of, and then that
Mm-hmm.
that reflects the light, you know, to a way that
Probably
you can see it 1,000
probably acts as a
times.
lens in itself.
Yeah, exactly. It refracts the light. It bends the light so that you can take a look at it
So do you need
at
a
that level.
thousand to see bacteria? Is that where you
No, no,
start
no.
see? No.
Nope. You can see. I
So
mean, you could see bacteria at 40, really, if you have the eyes for it. But
uh,
but 400 is really where you do all your observation. It's like when you everything is done at 400x, really. For like
so
compost
on
teas and stuff.
the same 400 you could see a bacteria, and but wouldn't like a trichoderma would be huge
A
comparatively,
trichoderma would be huge. From a bacteria, yeah, to like a
or
single
nemat
bacteria.
or
I mean, it
nematode
depends. So
even.
so it depends. So it depends. I mean, bacteria. We have like our larger bacteria, which can be like lactobacillus, which there's tons and tons of species of each one of these types of bacteria, and some of them are very 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 different in size. So the answer really is it depends, on what it is that you're looking at.
So bacteria, I so they're are they help me out a little bit. I'm gonna show you my what I don't know. Are
Okay.
bacteria single celled?
Yeah, I mean they can chain together. They can
Okay.
create like chains and stuff like that. Like bacillus species will chain, and then like Bacillus subtilis, will group up in little groups of two, and they kind of like orbit each other. They're pretty cute. And then a lot of Bacillus species will like chain together, and you'll kind of have like this long elongation of single rod um, bacteria that like link together and almost form like a ladder kind of.
Can you
And
watch
so
them? Can you watch them divide?
you can, if you sit there and you stare at it long enough, if you
Well,
if you
they
want to
must
do that.
divide pretty fast, right? I mean, if they're doubling their population.
They do divide pretty quickly, but I mean, like, you're not just going to look under a microscope and just watch bacteria divide like quickly.
No.
You would I mean, if I were to do it, I would put a camera on it, and then time lapse it, and then watch it that way. But I definitely don't have the patience to, you know. So
Yeah, I've seen pictures of cells dividing, but it seems fast, but that could have been just sped up. I don't know.
yeah, I'm pretty sure all those are sped up. Yeah, because
Fascinating
yeah,
though, isn't it? Isn't
yeah.
that fascinating that
And
there's something molecule that can create itself? It's amazing. It's amazing if you think about biology, biology at a very fundamental level. It's magic.
yeah, definitely. Hey, but there was one thing I wanted to touch on that I kind of got cut off by my internet connection,
Go ahead.
about the protozoa consuming oxygen levels, or consuming
Oh
oxygen,
yes,
changing the dissolved oxygen levels.
I was about to
Um,
ask you too.
so the more ciliates, the more protozoa, you know, the lower your dissolved oxygen levels can be. And there is a point where there's too much protozoa and your solution becomes anaerobic. Um, I use a dissolved oxygen meter to actually identify that point, um, you know, which I so that there isn't really any guesswork, because you want protozoa in your soil. But a lot of the time, you'll see people, they'll look under a microscope and they'll see a ciliate and they'll be like, it's anaerobic, and they'll just, they won't use it or something like that. But you need to, well, what I do is use a dissolved oxygen meter to actually identify whether or not the solution has really gone anaerobic. Um, and that's something that people don't usually talk about, and I really want to normalize. I want to kind of get the use of dissolved oxygen meters in compost tea to be more used, because I think there's a big portion of compost tea making that's being ignored by not using a dissolved oxygen meter.
I like, and I have a question for you. There's
Mhm.
different methods of dissolving oxygen in teas. Air stones, Venturi pumps, just the vortex method where it's basically just spinning the water.
Mhm.
Have you played around with by testing it which methods work the best, and how do you dissolve the oxygen in your water, or provide the water with oxygen, supplement it?
So okay, so dissolved oxygen is affected by a few different variables. The biggest one is really the temperature of your water.
Temperature is huge.
Uh-huh. So as you have like warmer water, your dissolved oxygen levels will go down. And that's just kind of like like when you have, like, when you fill up your glass of water and it's warm and you'll have little air bubbles in it. It's all the oxygen kind of like coming out of it and then just rising to the top, because your warm water won't hold that oxygen. So you have the cold water that helps hold more oxygen. And that has nothing to do with your question, but that's important to know.
No, it does.
And
Actually, it really does. That's
and
a
yeah,
reason to
so
brew teas colder, right?
so yeah, so I mean, just really like 75. You want pretty
I
much everything
agree.
you want between 70 and 75, really, right? Um, so as far as like which method of aeration is the best, they all work just fine. And you're not really going to push water past its point that of dissolved oxygen that it can actually hold, without making it colder. And so you'll get to a certain level and it'll stay there. You can do whatever you want to try to make more air in that thing, and it's not going to do it, because that's just not how it works. Um, uh, but you mentioned these different methods of compost tea making, and I've sampled pretty much all of them.
Hmm.
And conical brewers,
Hmm.
not vortex, are the best compost teas I've ever,
Really?
that aren't spinning without a tea bag. So free floating amendments, um,
Greater
with just
surface
like a,
area.
yeah, absolutely. And so you yeah, that's it. Greater surface area. You're expanding into the solution more. You're expanding your microbial inoculants into the solution more, and so they have better access to their food sources, to their humates, to their whatever it is that you're feeding them. May it be sugars or polysaccharides. Um,
The kinetic theory of reactions, by the way.
The kinetic theory of reactions.
Yeah. You want to move in fast and orientate in a lot of ways because it's all basically geometry, you know,
Mhm.
puzzle pieces. It's really amazing at that level. But yeah, the faster things are moving and the more surface area, the better chances for reaction, right?
Yeah, yeah,
Anyways, I went
definitely.
off.
Yeah.
Keep going. I
No, but
interrupted.
that's right. No, that's exactly right. Yeah. I didn't
I always
know that that
suggest
word
not
was.
using a tea bag just because from a chemistry point of view, it's going to mix better without it.
Yeah,
Anyways,
I don't like using tea bags either. There's a time when you need it. If you don't have a conical brewer, you don't have access to a system that doesn't use or that doesn't need tea bags. Because if you have your organic matter, you're creating sediment on the bottom. You do create anaerobic conditions, and then when you have anaerobic conditions that are mixed with aerobic conditions, that's when you really get your pathogens. It either needs to be completely aerobic or completely anaerobic. Um, Otherwise, you're going to get your pathogens. I'm talking about anaerobic conditions creating these anaerobic pathogens. And then earlier on in the talk, we were talking about making anaerobic teas.
you'll come back in a second.
Okay, it's the Starlink. I'm sorry. Um,
Don't be sorry for what you can't control.
and thank you.
Good information, ladies and gentlemen. I love talking about teas.
The teas. The
Love talking about teas.
What was I talking about?
Talking about anaerobic conditions and
Oh, yeah, yeah, yeah.
I remember
So
this one.
so we were I had mentioned that, yeah, I do use anaerobic tea sometimes. So anaerobic conditions, they can brew pathogens, but it's the anaerobic conditions mixed with aerobic conditions that really are like what accelerate pathogenic growth. um, so when you make an anaerobic tea, you're adding your microbial inoculants, you're adding your food source, you're mixing it up, and it's really important to add lactic acid bacteria serum. You don't have to do this, but this helps um, outcompete bad anaerobic pathogens, bad anaerobic bacteria and fungi. Um, and then you let that sit for, usually you do 24 to 36 hours. If you don't have a microscope, um, what you're really looking for is, if you do have a microscope, is you when it when your food source is consumed, when your bacteria get to like a point of either not multiplying as quickly and then you use it, or they reach the proper density and you use it, or you see that the protozoa are starting to come up and then you use it, which protozoa won't come up in an anaerobic tea. So in an anaerobic tea, you kind of watch it until they stop multiplying super quickly. um, and that's like when your food source is consumed. And so you don't want to have too much food source because your bacteria counts will go through the roof, and then you invite other bad organisms and stuff. And if you're doing an aerobic tea, you'll see that that's when the protozoa show up, is when the bacterial counts are really, really high, and then the protozoa will multiply very, very quickly trying to consume all that bacteria, and then you get anaerobic conditions very fast. So it's important to not overfeed your teas. um, so so what I was trying to say is really the proper technique is to monitor your compost tea with a microscope periodically, like every couple of hours, until you see your multiplication stop, and then use it. Um, yeah.
No, that's great points because there's a superficial making tea. You get some water, you put air in it, you put some kind of sugar or starch in it, you put compost or worm castings, real basic teas, which that's how I first got into teas. And I did pre 2010. I started brewing teas, which was started
Mhm.
kind of fairly early. Um, but when you start digging into it a little bit, basically you have an incubator, and you got to be careful because because you can make bad things in the incubator. Like I'm not big on fresh poop, and a lot of people don't even think about it. But sometimes fresh poop has E. coli, and E. coli you can actually multiply it in your incubator under the right circumstances. So, and you're talking about temperatures. I believe the warmer you get above 80, there's more pathogens can be introduced and cause trouble. But the tea, you gotta, there you gotta be careful. And what's a general time frame? I know water quality, the hardness of the water, what's in the water, temperature, the air, those things, are all factors on the growth in the tea. As a rule of thumb, I'm usually like one. If I want an aerated nutrient solution, I'll just bubble it in and water it within an hour. If I want a tea, I usually let it go for 2, 3, maybe 4 days. But after that, I know you don't want to go 2 weeks, right? You don't want to go a week really. It starts to
No.
smell bad, and I
So
usually go by smells. You go by looking at it under a microscope. What's a good time frame? A rough, safe area? Do you think? Temperature, time.
so a lot of this depends on what you're feeding it, the quality of your compost is, the temperature. Like you said, usually 24 to 36 hours is what I shoot for. Anything after that, you're going to start to see your is the foam a myth?
See, that's
Um,
what
yeah,
before you go ahead. I shouldn't tell you bias you at all, but I always went by the foam. I watched the foam rise and fall, and after I brew it a few times, I kind of and am I wrong about the foam being an indication of the things breathing in the water? Let's get that out of the way. Help
the
me out.
foam is degrading or degraded protein cells. So
Okay,
so
it's
it is
not CO
not
two.
no.
Oh,
Mm-mm.
my bad.
And a lot of and a
Sorry,
lot of the
everybody.
time, and a lot of the time, if you have any kind of like saponin or like emulsifier, um, something like that, you can get foam really easily. And so, like if you add something like yucca, you're gonna have foam like crazy. It's gonna be like a bubble bath in your composting area. Um, but also, you see a lot of foam when people are using manures in their composties.
Hmm.
So people will add like cow manure or guanos or even chicken manure or something like that, and then they'll see all this foam. And the foam isn't bad, it's fine. But it doesn't necessarily mean that it's really good either. Um,
Because I always saw
really,
a correlation, between at first a lot of times you'll see foam almost immediately, and
mhm.
that's a kind of different thing, and then and then you'll see the foam
Excuse
grow,
me.
and then it'll actually die down, and I always just said, well, that's they're animals and they're breathing out CO2, and sorry everybody but I always qualify. I don't know this stuff for sure, but
So,
that's how I went
so
on. Is the foam
um,
my bad?
when you see the organisms kind of off gassing CO2 and stuff, you'll start seeing carbonation. And when you make like an activated effective microorganism solution, AEM, which is that lactobacillus, molasses, and water thing I was talking about earlier,
Hmm. Hmm.
if you leave it sealed, it'll off gas tons of CO2. And if your container doesn't explode, it'll be carbonated when you open it. um, and same like with the second fermentation of kombucha, where you do your first fermentation, then you add in a little bit of food source to your bacteria, and then you bottle it, and then the carbonation, off gases in there, and it's sealed, and then when you open it, it's carbonated. And so when there's like off gassing from organisms, it creates like a carbonation. Um, but no, that's not that. Sorry, that's not what's happening in a composting.
No, just the whole idea of things like fermentation are so powerful. Even the things we eat, and like you mentioned, kombucha, I think it's
Mhm.
called.
Computer, yeah.
Fascinating subjects about kind of again going back, our stomach biome and all the bacteria that's in our stomach that actually live with us and help us break down our food. Fascinating stuff that I think we should all delve into a little bit. What do you think?
Yeah, absolutely. Kombucha, I love kombucha. Can I talk a little bit about kombucha? How long do you usually have these talks for?
I usually go about 2 hours because I don't want to
Two.
punish
Okay.
punish my viewers. But
Okay.
tonight's been I've gone 3, 4. We'll go whatever. No, I
Okay.
would love to have you come back on because I can go down some rabbit holes. But I
Great.
love talking tea. I love talking tea. So let's hear about kombucha.
Yeah, definitely. So, I love making kombucha. I really like making it with red beet and with like lavender and sage and different like herbs and stuff. I use kombucha to treat powder mildew, to fight against powder mildew.
Oh, really?
Um, yeah, and it works fantastically. and a lot of people don't believe me. And there was even some people who talked about kombucha and who tried to replicate an experiment that I did, because I did an experiment with kombucha and I posted it in the Probiotic Farmers Alliance. And some people tried to replicate it and they couldn't do it, and they were like, "Oh, it just doesn't fucking work. She lied or some shit." And but one of the things that people missed, because they tried to look at all the different variables, being enzymes or pH and stuff. But what I found was like the most important component, in addition to enzymes and pH, which absolutely have an effect, is the carbonation level of kombucha when spraying it on your leaf surface. And that carbonation actually will agitate, the surface of the leaf and like reacts to it, or not reacts to it, but it agitates that PM, so that the enzymes and the pH can do a better job at killing it, at getting rid of it. So I use things that create high carbonation, like red beet, which is like a prebiotic. It'll feed microbes. It's a really great microbe food. And I don't want to speak for Alan Atkinson, but I know that he uses red beet juice in his bukashi making processes, and I use red beet when I'm in my watering sometimes, just giving it to my plants, just because it's such a good, bacterial food. It's a good prebiotic. So I use red beet when making kombucha that I use to treat for powder mildew, which I haven't gotten powder mildew in forever, but when I did, that's what I would do, and it worked really, really well. And if you have problems with pH or PM, I recommend trying it.
As a preventive measure, or once you get it?
Once you get it. So it's really good just for plant health. Adding kombucha and that lactic acid bacteria serum that I was talking about, labs, it's a KNF input. I mix 15 milliliters per gallon of labs, and then an ounce, 30 milliliters per gallon approximately, of kombucha to a foliar spray. And I do that pretty regularly. You don't really like soak your leaf. You don't want it dripping. You just want to kind of like mist it, and just like very lightly. And you see like a great like health.
And would you agree that it's always best to foliar feed it in the dark cycle? I've heard that a lot. You'll come back. Cheers. I'm seeing some great little sideshow in the comments. I'm loving the information. We got her back again. So another little known thing about this KNF stuff, when we're talking about ferments and the, like that you were talking about the cheese making process, almost all of this humans can eat too, correct? And it's beneficial for humans, right? Like even the kombucha, do you drink your own red beet juice kombucha, or is it just nasty?
No, definitely. Yeah, I love my red beet. It's really good. No, most of the stuff that I use for my plants, I actually ingest it. Like I have a mushroom complex that I take and that I put in my smoothies and stuff like that, which is I don't even really know the proper pronunciation. So there's the reishi and the shiitake and the maitake, the chaga, the cordyceps, and turkey tail, I think. It's like a mushroom complex that I take, but I also give it to my plants. And it's really rich in beta glucans, which is a polysaccharide. And that helps feed biology and stuff like that. It helps feed more diversity of biology.
Without going too much into a tangent, up in Oregon, you guys got some new rules where you can actually do psychedelic mushrooms a little bit now, right?
Yeah, definitely. Yeah.
We won't talk about that. We'll save that for another show.
Okay, that sounds good. Yeah, I haven't
No,
eaten mushrooms in a long time, but
no, all those areas fascinate me, and there was I grew up in the time where they cracked down on everything because of the Vietnam War protesters. They they did drugs and they protested against the war. This is my kind of take on it. So they shut down the drug thing when I was a kid. And now they're finally starting to. It was a crazy time if you think about it. The 70s until now, for a 50-year period where draconian, and it still is to some extent, but this whole community with things like microdosing and mushrooms and probiotics and KNF. I love being in this. To me, it's cutting edge stuff, but it's really
Oh, I
ancient.
love it. It's so
So
great. I love that. I love that about it. It's like this like almost kind of esoteric, like awesome information that is, yeah, very.
oh darn it, just in time. Good show tonight. I think. I definitely would. Man, I can talk tea. I can talk turkey tea too. Turkey mushroom tea. Turkey tail. Anyways, maybe she's gone. Maybe she'll come back. We'll see. So how's it going out there? You guys getting your plants started? I'm gonna start popping my seeds this weekend. Am I a little too late? I don't remember. I think I was late last year. But again, at the end of the year, 12, 15 foot plants. I'm pretty sure. So I don't know if you have to start real early, but I think I'm going to this year. I got a nice bunch of seeds that I'm going to try out. Kind of excited. It's always fun to try out different strains. I got a lot of, most all crosses I get from generous donors who may be listening. Hey, Daniel Jackson. Good to see you up in here. Or you must be in a medical state. I don't know. It's a little bit sketchy to ask people where they're, because some states, like I was saying, it's still rough. And I think, what was it, Utah recently said they were going to put it in their constitution to never let cannabis be legal, which is just nuts. What's going on? What's going on with these people out there? I thought we were trying to get in a little bit of an enlightenment period, which has been fun to live through. A lot of people who've come on this show from Humboldt County, because it's kind of Humboldt centric, were around in the 90s when they had Black Hawk helicopters come with people in black who came down off ladders and came and stormed your farm, and you went to prison or jail for sure and took everything you had. And that wasn't too far in our history. And then we got the medical here in California, I think around 96. And that was kind of a golden area for growing. That was a golden era. Very profitable. Actually, before then was too, but then the growth started coming out and all the products started coming out on the market, and it exploded. And now we're getting another explosion in technology too once recreational hit. I've seen that with lighting technology and learning about KNF and compost teas and really cool stuff going on. So it looks like we might have lost her. So here we go. This is the stuff I'm talking about here. That's just crazy. See, that's just crazy. Dirt road dude. That's a shame. Yeah, there's something. Actually, I have a customer in Mexico, and I feel bad for the guy for a number of reasons. But he's in the middle of the government and cartels, and he's in a weird area. At least I hope he's not in a cartel. But no, in Mexico, I think Mexico's kind of like they say all drugs are legal in Oregon. I think all drugs. I don't want to say legal, but I don't think you go to jail for them. Like even heroin or cocaine or methamphetamine, I don't think you go to jail. I think you go to fine and you have to go to maybe a drug rehab thing or something. I'm not real sure what the technicality is, but it's a little misinformed to say, oh, they just legalized all drugs in Oregon. I just think they decriminalized them, which is a different kind of thing. And I think it's the same in Mexico now. I'm not sure. Well, we got her back again. Thank goodness. Let's see if we can get another 10 minutes or so out of this young lady. Fascinating talk.
Great.
And thank you for being such a champ and getting knocked down and coming back up again.
Yeah, no worries. That time my phone died, so.
You're a trooper.
Um,
So where were we at? Ben, please come back on again sometime,
yeah, for
if
sure.
you would.
I'd love to.
I would love to have
Yeah.
you and Wendy come on, and maybe, I don't know if you know Pearl Moon. She's an interesting lady.
Mm-mm.
She's out of Southern Humboldt, real old school grower, pioneer. I love the kind of I love to hear people's story and how they got in this industry. which
Yeah.
we touched upon a little bit with you. If you don't mind me going back, you said you grew in college. Was that indoor growing then? Were you doing that in
Yeah.
college?
So I grew up in California, um, and I moved out of my parents' house. When I was 18, I moved to Lake Tahoe.
Hmm.
So that's kind of barely Northern
From
California.
where in California? Let's go back a little bit.
So I was in SoCal. I was in like the I guess like the Long Beach area.
Okay.
Um, yeah, and then moved up to Lake Tahoe. And Lake Tahoe, all we did was snowboard and smoke weed and go to the beach. It was pretty fucking awesome. And but in South Lake Tahoe, there aren't very many good places to grow outdoor. Everyone's growing indoor. There's a lot of indoor growers up there. So that's kind of what I got submerged into, was like the indoor growing culture of South Lake Tahoe. And people were growing fire indoor up there. The water's really good from Lake Tahoe. Um, so that's really how I got started. Yeah, growing indoor medically.
So did you go to college in that area?
Yeah.
Or- okay.
Yeah.
University of Nevada?
No, I went to LTCC, and then I transferred when I moved to Chico, and then I
Oh,
finished up in Chico.
I went to Chico High School.
Oh, cool. Yeah,
I
I
was
lived in
born
Chico
at,
for 10 years.
you probably heard of Fontana, I'm sure.
This sounds familiar.
San Bernardino, Fontana.
Oh, okay. Yeah,
The,
yeah, yeah.
the best parts of Southern California, that's where I grew up.
Okay, cool.
Yeah,
Yeah,
I'm familiar. Yeah. I got my teaching credential at Chico State too.
oh, really? Cool.
Hmm. Hmm.
That's great. Yeah, I went there and did my business management thing.
Well, that's a really neat little town too. Actually, that's where I learned I, I went there in the middle '70s. Um, one of my aunts was one of the first growers up in the Oroville area, and so I've
Oh,
been
yeah.
around this industry since '75 or so. I used to. Can I say I sold weed in high school? I
Yeah.
did.
Uh-huh.
I guess
No, I
I
did.
guess
I
the
did too. It's okay.
statutes of limitations I'm sure have run out by now. But yeah, but I used to sell my family's weed in high school. You had quite top market for it. But anyways,
Yeah, I
and
I bent some rules back then too. It's okay. It's all
I
good.
think that's what's
Okay.
interesting about our industry is kind of by definition you had to be a rule. You can't take rules real seriously. Otherwise we'd never get into this, right?
Yeah.
So
Yeah.
a little
I
bit
don't
of
know.
rebels, and that's kind
I
of a
don't
neat
know.
personality trait.
Yeah,
You have to
for
be
sure.
rebels.
We're definitely kind of a counterculture community that's been pushed into the mainstream. You know.
But then,
So
once it became recreational and more legal, I'm sure you've seen this too, an influx of money, and they have a different mindset of
oh
profit
yeah.
and
I mean, they treat the plant like a, you know,
a
like a
commodity.
yeah, like
They
just
don't
a way to
worship it.
get rich. No, definitely not,
Yeah.
And they build their companies without prioritizing the passion for the plant, and that's why they don't last long and why their weed sucks. So, and
That's some of the
it's
good and bad about the green rush. Everyone thinks they're, going to come here and make $1 million, but it's farming, and farming's not. It's not easy.
absolutely.
It's relying on things that are out of your control, which can be heartbreaking sometimes. Darn it. We're gonna have to get you a new. We're gonna have to get you a new internet service provider. Anyways,
Sorry, I dropped again.
well, that's all right. Um,
Yeah.
hey, if you're man, I don't. If you're ever on your way down to Humboldt, I would love to have a cup of coffee with you. But I would love for you also to come on the show again because We've gone about 2 hours, isn't it? I love these conversations.
Yeah.
Because
Yeah,
they're easy.
it's been about an hour and 53 minutes.
Pretty
Um,
darn close. So anything else you want to get, any other, because there's a lot of points. We could talk all night. It's just
I'm sure.
my
Um,
mom's watching and she needs to go to bed.
okay. Sorry, Mom. I don't know. Is there any like questions maybe like
Oh, good,
maybe a
good,
couple
good,
quick
good.
questions
Excellent
or
idea.
something?
I can flash them up on the screen too if there's
Sure.
a good one.
Sure.
But yeah, if anybody have any questions for Luna, great. I don't know. Can you see the comments?
I saw the last one that popped up. I don't see
Yeah,
the
but
comments, but I saw that question, the last one that popped up.
yeah, I can. There, I think there's a way. Depends what platform you're on that you can actually you can see what they're typing into. There might be a little button you can push or something. I don't know. It says comments or
Oh,
something like that.
chat, private chat, comments. Oh
We've had
wow.
actually
Okay, now I see them.
okay. We have actually had a pretty good show tonight. We get people from Canada, Puerto Rico, Mexico. It's pretty. It's kind of this technology is, even though it drops out once in a while. That's really common. So don't feel, that's really common in this that we have these little hiccups. But
Yeah,
it's worth
no
it.
worries. Yeah,
No worries.
I just switched to from my, I live out in like this kind of, and so I was on crappy satellite internet, and so I got Starlink, and it's a lot better, but it's still in beta, so it's dropping all the time. I don't really see any questions. It's okay.
No, I haven't seen it.
Okay.
Are you familiar with? Are you in the Wolf Creek area by any chance?
No,
No.
no, I don't want to give out my exact location if that's
Okay,
okay.
you don't have to give out.
Yeah.
Have you heard of the place called Green Source Gardens? They're outside of Medford, I believe.
I have heard of them, yeah.
I've done that's that's a very interesting guy who does it. They're one of the this is years ago. This is 10 years ago. They were the guy wanted to do everything local. I mean, within his boundaries, and he did a great job. But you'd be walking through the rows of plants, and you would see a goat skull. A goat skull just coming out of the ground. He would just bury his animals. It was you do.
Sure.
I learned about hugel culture, but
Yeah.
and I think he does seminars too. So if anybody's listening, they really want to like say, and I don't know if he's into KNF. He might be. He might be. I don't know what was going on there, but no, that's a real interesting farm if you ever get a chance to visit. And I think there's one out in Wolf Creek area, which I don't know where it is.
I've heard some cool things about Wolf Creek. I drove through there maybe a couple months ago, but no, I don't.
Yeah,
I don't
you can
live
drive
out
through
there.
Southern Oregon if you're from a non-wheat growing state, and you would be amazed about all the plants you will see along the
Yeah,
highway.
just going up the I-5. Yeah, you can just see plants from the highway.
Oh, everywhere. Yeah, it's
Yeah,
crazy.
everywhere. Yeah, they're all over the place. Go drive through Williams,
Okay, let's see here. Seriously great guest. I would love to hear more about kombucha foliar. There you go.
Sure.
That's a quick one. Yeah, we did. I don't know
Sure.
if you finished if you finished your thoughts up on that, but basically, I don't know if he gave you a recipe.
Oh, yeah. I'll walk through how to make kombucha for plants.
Good idea.
You can drink it too. So when you're making kombucha for plants, you want your Brix levels as low as possible. So your sugar content as low as possible. So when I make my kombucha, I have a Brix meter. It's a refractometer. So I measure the sugar. Yes, everyone needs a refractometer. Everyone needs a dissolved oxygen meter, a pH pen. Anyway, so you to make your kombucha, you start with black tea and your SCOBY.
What do you mean black tea? Let's start again. Black tea, just like tea that you buy black tea from a store?
Yep.
Okay.
Yeah. So black tea, the SCOBY, be good if you could get some from a friend who already makes it. That's going to be best from your local area. You can order it online if you really have no other options. Black
is that
tea,
the starter
SCOBY.
Source? The SCOBY? That's a new word to me. Is that the starter?
So SCOBY is a acronym actually. It's a symbiotic culture of bacteria and yeast. And it's essentially a big pancake looking thing that floats on the top of your kombucha. And it's the source of your bacteria, and it's mostly lactobacillus and yeast. And so you have your SCOBY that sits on top, your black tea. I'm just going to give you the recipe for a gallon because it's really simple. So for 1 gallon, you have 1 cup of.
Oh, what a time to drop out. we're on pins and needles here. So a gallon of black tea, some SCOBY, and then we're going to have to start back from there.
Sorry. And then your
Okay, we got a gallon of black tea. That's where we lost you.
that's right. Gallon of black tea, your SCOBY, a cup of organic sugar, unbleached, raw sugar. Don't use molasses; it'll clog your SCOBY. Don't use brown sugar; it has molasses in it. It'll clog your SCOBY. And some people are really stubborn that they just want to use molasses anyway. Your SCOBY last as long, and it will die. So just regular white cane sugar. Cut that for your gallon of black tea. And then I forget how, I think it's like 8 tea bags or something like that for a gallon of black tea. I would just follow whatever the instructions on the back of whatever tea you buy is, because sometimes they have different amounts of black tea. So you let that ferment. It's an aerobic ferment.
How long?
So, I mean, I monitor it with my Brix meter. I watch it, and I check my pH. So when the pH drops below like 4, is when I start to use it. And when the sugar is all consumed, I start to use it. And so if you don't have these instruments, 2 weeks is probably safe, Making kombucha is pretty simple. You can always look online. There's like kombucha guides and stuff like that. So once you have that, your first step of kombucha, that's called an like an F1 even, but not to get that confused with like the genetic F1. Yeah. And then once you have that, you bottle it. So you take an aerobic ferment, you put it in a bottle, and then you add a little bit of food source to it. And this is where all the different flavors come from and stuff. So you can add like red beet juice, or you can just stuff it full of lavender, which is what I love to do, or rosemary or sage. And you can play with the potency, whatever. And then the bacteria, the yeast, will consume some of the things inside of the organic matter that you put in there. And then you have nice carbonation. Oh, and you bottle it. Sorry, you put the lid on it. You seal it. You air seal it, and you let it ferment for like 3 days. Let it burp, because if it has a lot of carbonation and you don't burp it, it can explode. And it's like a little bomb throwing glass shrapnel all over your house, and it can be bad. So when you're doing it,
so to use a bottle that has like a cork that could remove on it, or
they have like like those. So when I use it, they're like glass bottles. They have like a rubber grommet and like a plastic top, and then like those metal little clippy things.
The clamp.
Yeah, the clamp. So that's what I use. And then you can kind of just like lightly off-gas it, so it doesn't just throw kombucha all over the place. So I burp it at least one time, and then if you burp it, kind of see how much it's off-gassing. If it's not off-gassing much, stop burping it. You want it to carbonate, because you really want that carbonation. If you're really looking for carbonation, I use the red beet when I'm making it for tree powder mildew, because it's like it gets really, really carbonated. And even if you go to the store and you buy red beet kombucha, they're usually the ones that when you open them, they spray all over the place, because they just are so prone to over-carbonating and to carbonating. So I'll add about a tablespoon of red beet juice to the bottle.
Okay. And for the plant, do you put the lavender in there, or you just use the red beet juice? For the plant, I go with the carbonation, so I use the red beet juice. But I love the lavender and the sage and the rosemary to consume, and you can also spray that on your plants. You just want to make sure that your Brix level before you spray it on your plants is super, super low. You don't want any sugars. If you have high sugar content in your kombucha and you spray it all over your plants, you're gonna be really, really sad. So I've seen people, they pay attention to the sugar content level, and they spray their plants with it, and they got like white flies, that were attracted to the sugar. They got like infestations and stuff like that. So
So this is, if you want to save the money, this is how you do it. I mean, it's really, really effective. You just got to be careful because you don't want to spray sugar all over your leaves. And if you do, and that happens, you can spray labs, the lactobacillus serum, and that'll help eat up a lot of that glucose, a lot of that sugar too. So I just usually, out of precaution, and because it has enzymes and stuff, I add
The final ingredient we missed. Wouldn't you know it? She'll come back, I'm pretty sure, finish this thought if she can. But we talk once in a while about like new growers or what are some of the equipment you should use, or it could be endless with monitoring. And she's talked about a Brix meter, which is I'm going to just talk about this real quick because I think it's one of the essential that's rarely talked about. And it measures it measures basically the sugar content in a liquid, and it's a real simple device. It's a passive device. It's a prism, and there's a little gradient scale on there, and you look through like a telescope, and you put it in there and hold it up to a light source, and it gives you a number. I can't tell you what the number is right now, but it measures sugar. So when you're measuring the kombucha, you don't want much sugar, but if you want to know how healthy your plants are, you want a lot of sugar. Like take a leaf and squeeze the juice out of it and put it on there. And the higher the Brix level, usually that's a very indication of how healthy the plant is, correct? Because that's how well photosynthesis is making that sugar, right? So
that's right.
If that's going good, you're going to have a lot of sugar. So it's an excellent, and it's what, $50, $60 for a Brix meter?
Yeah, something like that. Yeah.
Something like it's very reasonable, but that's one of the first tools you should get, and they're rarely talked about. So I'm glad you brought up a Brix meter. Having I hit the wrong button somewhere. Anyways, I'm
Sorry,
back.
You disappeared
That
for a
was
second.
me. That was me. Um, so yes, I went on for a little tangent, but yeah, Brix meter, a good tool to have, and for all kinds of stuff. Go ahead and finish your thoughts on, and I'll try not to interrupt.
Oh no, it's cool.
I know it's cool.
But so the Brix meter, I don't even know what I was talking about, but it was cool what you were mentioning about testing like the sap, the juice of your cannabis leaf for the Brix content, and the higher it is, the healthier it is, and that has to do with like micronutrient absorption and carbohydrate production, just like general vigor. I've gotten mine up to 24, the highest I
Yes,
got it.
I'm not sure what the numbers are, but they're relative. It's a good idea to start playing around and checking what a healthy plant looks like, what a bad plant looks like, and you kind of get a range. But it's a really good tool to measure the health of your plant. And something I want to touch on too for beginners. And I'm sorry, I said I wasn't going to do this, but I see a lot of forums, and actually there's an inside joke you've probably seen. It's like you need CalMag, right? When you're dealing with living soils and you're working in living soil and it's really healthy, nutrients aren't the issue. I mean, rarely are nutrients the issue if you have a good living soil. It's going to be your environment or a pest or a pet, mold or something that comes in. Those are going to be your issues if you're doing things right. And most people go right to I don't have enough of something. First of all, you probably have too much of something. That's where your problems come in. Probably water too much or something. But it's an environmental issue. It's not, and that allowed me to advance. So not thinking what nutrient am I missing, because the plant kind of takes care of that if you got a good healthy living soil. And I went off on that tangent, but I think that's so important. It eliminates that. What I need CalMag. You don't. You really
No,
don't.
usually you don't. So if people comment CalMag in a lot of the groups that I'm in for it, I'll kick them out. And I don't know, that may be kind of mean, but like I just have zero tolerance for that kind of crap anymore. Like in an organic group, if people recommend to like buy a bottle of CalMag, I'll just kick them out of the group. it makes me so mad. But there's so many causes of uptake issues, which isn't a soil nutrient deficiency. It's that your plant isn't getting that nutrient. And so instead of asking what are all the compounds present in my soil, why is this nutrient being uptaken, they're just like, oh, it just means I need to add more. I'm just going to pump it with whatever because I see deficiency, so I'm just going to add more of that and hope that it goes in. When really the issue it can be you're overwatering, or your biology is fucked up. You have an infection. It's too cold. People, a lot of the time I see people put their plants straight on their concrete in their garage or something like that.
Idea. Yes, bad idea.
Did you hear what I said?
I got to the concrete is a bad idea too.
Yeah.
Yes. Yeah. Roots like a a little bit warmer. They don't want the heat drained out of them.
Yeah, exactly. And then they'll have that calcium uptake issue because calcium's always the first to go, and then they just start wanting to pump it with calcium, and then people start recommending CalMag, and it's like, dude, your plant is sitting on the concrete and it's really cold. You should put it on a pallet or a box or a crate or get a table, maybe. So.
Okay, maybe you can clear up this because this goes back. Okay, this will be the last one question I'm going to ask you. I think.
Okay.
Maybe not. Don't hold me to
okay.
that. Water into roots. Now, I looked into it a little bit, and I tell people this, so I apologize if I was lying to you. But what drives the water molecule going into the root is one leaving the bottom of a leaf on the stomata. That's what pulls it. It actually pulls it in from the top. Is from my understanding. That's probably one of the mechanisms for a plant to absorb water. It needs to release water out of a leaf. So that's why I think it's an environmental issue. You definitely want airflow, low humidity airflow underneath a leaf. That's why I think airflow is a key to a healthy plant. It's going to drink faster if it can evaporate faster. Am I wrong to give that advice?
No, not at all. That's definitely on point. Are you familiar with vapor pressure deficit?
No.
No.
But I like those big words. Tell us.
Them big words. So vapor pressure deficit is like a scale. So a lot of the time, people talk about humidity, relative humidity, but they don't understand that relative humidity is not a representation of how much vapor is in the air. It's a representation of how much vapor is in the air in relation to the temperature that it is. Mm-hmm. So, if it's 100 degrees and 100% humidity, there's a lot of vapor. There's a lot of moisture, water in the air, because heat can really keep the moisture in the air. And if it's cold and it's 100% humidity, there's much, and they're both 100% humidity, but there's way less water in the air. So, yes. So the vapor pressure deficit, the vapor pressure scale, is really what needs to be paid attention to. So the airflow is definitely key to it, because you want fresh carbon dioxide being brought in, fresh air circulation. Things don't stagnate. It all needs to breathe. Soil needs to breathe. Soil needs the oxygen. Leaves need the carbon dioxide. But also staying on top of the proper vapor pressure deficit is really important also. And there's a little scale, if you just look at vapor pressure deficit, or VPD, that shows based on what temperature it is, what humidity it should be for your plant. Just look it up in relation to plants. And yeah.
That'd be more pertinent for the indoor grower, because outdoor, I think there's a lot wider range of what works outdoors is a lot wider range than what works indoors. You can't have a grow room at 100 degrees, but you can grow outside at 100 degrees. But I'm sorry, it's not going to work indoor. There's a different buffering, or there's a lot more forgiving outdoors. I really believe that. So yeah, when you're talking about that, those are the things you can control indoors, somewhat to extent in greenhouses at some level. You can localize the humidity and stuff a little bit, but
yeah, definitely. Indoor. Yeah, definitely indoor. Yeah.
Well, I've taken up over 2 hours of your time, of your life, and that's not a large percentage. It's more larger percentage for you than me at some level. So anyways, Great, fantastic talk, and we could go on, and I just can't do this to my mom and my bladder.
It's all good. Sorry,
We're
sorry,
getting
Mom.
old.
It was great talking to you, and yeah, let's do it again sometime.
I would love to, and let's just sign off, and because I'll just go on tangents, and I don't want to do that. Thank you for
Mm,
coming in, I always love to see get. I saw somebody from a modern genetics maybe. Love to have some people in the industry come on. So just leave some comments if you want to come on the show and talk. I do every other show is usually open panel. Anyone can come on, but or if you know anybody, Luna, that would like to come on, that's interesting. I love people who have a story to tell in their journey in cannabis, and
sure.
awesome people come on the show. So I really like it. But other than that, let's just say goodnight because I take too long doing this. I'm thank you so much for coming on, giving your time and fighting through the mishaps and not just giving up. And
Absolutely.
thumbs up.
Thanks so much for having me.
You get a good night's sleep and have a great weekend.
All right, you too.
All right. Good night, everybody.
Bye.
Good show.